Gel electrophoresis is a method for separation and analysis of macromolecules and their fragments, based on their size and charge. It is used in clinical chemistry to separate proteins by charge and/or size, and in biochemistry and molecular biology to separate a mixed population of DNA and RNA fragments by length, to estimate the size of DNA and RNA fragments, or to separate proteins by charge. Nucleic acid molecules are separated by applying an electric field to move the negatively charged molecules through a matrix of agarose or other substances. Shorter molecules move faster and migrate farther than longer ones because shorter molecules migrate more easily through the pores of the gel. This phenomenon is called sieving. Proteins are separated by charge in agarose because the pores of the gel are too large to sieve proteins. Gel electrophoresis can also be used for separation of nanoparticles. Gel electrophoresis uses a gel as an anti-convective medium and/or sieving medium during electrophoresis, the movement of a charged particle in an electrical field. Gels suppress the thermal convection caused by application of the electric field, and can also act as a sieving medium, retarding the passage of molecules. Gels can also simply serve to maintain the finished separation, so that a post-electrophoresis stain can be applied. DNA gel electrophoresis is usually performed for analytical purposes, often after amplification of DNA via PCR, but may be used as a preparative technique prior to use of other methods such as mass spectrometry, RFLP, PCR, cloning, DNA sequencing, or Southern blotting for further characterization. Physical basis. In simple terms, electrophoresis is a process which enables the sorting of molecules based on size. Using an electric field, molecules can be made to move through a gel made of agar or polyacrylamide. The electric field consists of a negative charge at one end which pushes the molecules through the gel, and a positive charge at the other end that pulls the molecules through the gel. The molecules being sorted are dispensed into a well in the gel material. The gel is placed in an electrophoresis chamber, which is then connected to a power source. When the electric current is applied, the larger molecules move more slowly through the gel while the smaller molecules move faster. The different-sized molecules form distinct bands on the gel. The term gel in this instance refers to the matrix used to contain, then separate the target molecules. In most cases, the gel is a cross-linked polymer whose composition and porosity is chosen based on the specific weight and composition of the target to be analyzed. When separating proteins or small nucleic acids, the gel is usually composed of different concentrations of acrylamide and a cross-linker, producing different-sized mesh networks of polyacrylamide. When separating larger nucleic acids, the preferred matrix is purified agarose. In both cases, the gel forms a solid, yet porous matrix. Acrylamide, in contrast to polyacrylamide, is a neurotoxin and must be handled using appropriate safety precautions to avoid poisoning. Agarose is composed of long and branched chains of uncharged carbohydrate without cross-links, resulting in a gel with large pores allowing for the separation of macromolecules and macromolecular complexes. Electrophoresis refers to the electromotive force that is used to move the molecules through the gel matrix. By placing the molecules in wells in the gel and applying an electric field, the molecules will move through the matrix at different rates, determined largely by their mass when the charge-to-mass ratio of all species is uniform. However, when charges are not all uniform, then the electrical field generated by the electrophoresis procedure will affect the species that have different charges and therefore will attract the species according to their charges being the opposite. Species that are positively charged will migrate towards the cathode, which is negatively charged. If the species are negatively charged, they will migrate towards the positively charged anode. If several samples have been loaded into adjacent wells in the gel, they will run parallel in individual lanes. Depending on the number of different molecules, each lane shows separation of the components from the original mixture as one or more distinct bands, one band per component. Incomplete separation of the components can lead to overlapping bands, or to indistinguishable smears representing multiple unresolved components. Bands in different lanes that end up at the same distance from the top contain molecules that pass through the gel with the same speed, which usually means they are approximately the same size. there are molecular weight size markers available that contain a mixture of molecules of known sizes. If such a marker was run on one lane in the gel parallel to the unknown samples, the bands observed can be compared to those of the unknown in order to determine their size. The distance a band travels is approximately inversely proportional to the logarithm of the size of the molecule. There are limits to electrophoretic techniques. Since passing current through a gel causes heating, gels may melt during electrophoresis. Electrophoresis is performed in buffer solutions to reduce pH changes due to the electric field, which is important because the charge of DNA and RNA depends on pH, but running for too long can exhaust the buffering capacity of the solution. Further, different preparations of genetic material may not migrate consistently with each other, for morphological or other reasons. Types of gel. The types of gel most typically used are agarose and polyacrylamide gels. Each type of gel is well suited to different types and sizes of analyte. Polyacrylamide gels are usually used for proteins, and have very high resolving power for small fragments of DNA. Agarose gels on the other hand have lower resolving power for DNA but have greater range of separation, and are therefore used for DNA fragments of usually 50 to 20,000 BP in size, but resolution of over 6 megabytes is possible with pulsed field gel electrophoresis. Polyacrylamide gels are run in a vertical configuration while agarose gels are typically run horizontally in a submarine mode. They also differ in their casting methodology, as agarose sets thermally, while polyacrylamide forms in a chemical polymerization reaction. Agarose. Agarose gels are easily cast and handled compared to other matrices, because the gel setting is a physical rather than chemical change. Samples are also easily recovered. After the experiment is finished, the resulting gel can be stored in a plastic bag in a refrigerator. Agarose gels do not have a uniform pore size, but are optimal for electrophoresis of proteins that are larger than 200 kDa. Agarose gel electrophoresis can also be used for the separation of DNA fragments ranging from 50 base pair to several megabases, the largest of which requires specialized apparatus. The distance between DNA bands of different lengths is influenced by the percent agarose in the gel, with higher percentages requiring longer run times, sometimes days. Instead, high percentage agarose gels should be run with a pulsed field electrophoresis, or field inversion electrophoresis. Most agarose gels are made with between 0.7% and 2% agarose dissolved in electrophoresis buffer. Up to 3% can be used for separating very tiny fragments, but a vertical polyacrylamide gel is more appropriate in this case. Low percentage gels are very weak and may break when you try to lift them. High percentage gels are often brittle and do not set evenly. 1% gels are common for many applications. Polyacrylamide. Polyacrylamide gel electrophoresis is used for separating proteins ranging in size from 5 to 2000 kDa due to the uniform pore size provided by the polyacrylamide gel. Pore size is controlled by modulating the concentrations of acrylamide and bisacrylamide powder used in creating a gel. Care must be used when creating this type of gel, as acrylamide is a potent neurotoxin in its liquid and powdered forms. Traditional DNA sequencing techniques such as Magsam-Gilbert or Sanger methods used polyacrylamide gels to separate DNA fragments differing by a single base pair in length so the sequence could be read. Most modern DNA separation methods now use agarose gels, except for particularly small DNA fragments. It is currently most often used in the field of immunology and protein analysis, often used to separate different proteins or isoforms of the same protein into separate bands. These can be transferred onto a nitrocellulose or PVDF membrane to be probed with antibodies and corresponding markers, such as in a Western blot. Typically resolving gels are made in 6%, 8%, 10%, 12% or 15%. Stacking gel is poured on top of the resolving gel and a gel comb is inserted. The percentage chosen depends on the size of the protein that one wishes to identify or probe in the sample. The smaller the known weight, the higher the percentage that should be used. Changes on the buffer system of the gel can help to further resolve proteins of very small sizes. Starch. Partially hydrolyzed potato starch makes for another non-toxic medium for protein electrophoresis. The gels are slightly more opaque than acrylamide or agarose. Non-denatured proteins can be separated according to charge and size. They are visualized using naphthol black or amido black staining. Typical starch gel concentrations are 5% to 10%. Gel conditions, denaturing. Denaturing gels are run under conditions that disrupt the natural structure of the analyte, causing it to unfold into a linear chain. Thus, the mobility of each macromolecule depends only on its linear length and its mass-to-charge ratio. Thus, the secondary, tertiary, and quaternary levels of biomolecular structure are disrupted, leaving only the primary structure to be analyzed. Nucleic acids are often denatured by including urea in the buffer, while proteins are denatured using sodium dodecyl sulfate, usually as part of the SDS page process. For full denaturation of proteins, it is also necessary to reduce the covalent disulfide bonds that stabilize their tertiary and quaternary structure, a method called reducing page. Reducing conditions are usually maintained by the addition of beta-maratoethanol or dithethritol. For general analysis of protein samples, reducing page is the most common form of protein electrophoresis. Denaturing conditions are necessary for proper estimation of molecular weight of RNA. RNA is able to form more intramolecular interactions than DNA, which may result in change of its electrophoretic mobility. Urea, DMSO and glyoxal are the most often used denaturing agents to disrupt RNA structure. Originally, highly toxic methylmercury hydroxide was often used in denaturing RNA electrophoresis, but it may be method of choice for some samples. Denaturing gel electrophoresis is used in the DNA and RNA banding pattern-based methods DGGE, TGGE, and TTGE. Native gels are run in non-denaturing conditions, so that the analyte's natural structure is maintained. This allows the physical size of the folded or assembled complex to affect the mobility, allowing for analysis of all four levels of the biomolecular structure. For biological samples, detergents are used only to the extent that they are necessary to lyse lipid membranes in the cell. Complexes remain for the most part associated and folded as they would be in the cell. One downside, however, is that complexes may not separate cleanly or predictably, as it is difficult to predict how the molecule's shape and size will affect its mobility. Unlike denaturing methods, native gel electrophoresis does not use a charged denaturing agent. The molecules being separated therefore differ not only in molecular mass and intrinsic charge, but also the cross-sectional area, and thus experience different electrophoretic forces dependent on the shape of the overall structure. For proteins, since they remain in the native state, they may be visualized not only by general protein staining reagents but also by specific enzyme-linked staining. Native gel electrophoresis is typically used in proteomics and metabolomics. However, native SDS page is also used to scan genes for unknown mutations as in single-strand conformation polymorphism. Buffers. The buffers in gel electrophoresis are used to provide ions that carry a current and to maintain the pH at a relatively constant value. There are a number of buffers used for electrophoresis. The most common being, for nucleic acids tris-EDTA. Many other buffers have been proposed, for example lithium borate, which is almost never used, based on PubMed citations, isoelectric histidine, pK-matched goods buffers, etc. In most cases the purported rationale is lower current and or matched ion mobilities, which leads to longer buffer life. Borate is problematic. Borate can polymerize, and or interact with sidiols such as those found in RNA. TAE has the lowest buffering capacity but provides the best resolution for larger DNA. This means a lower voltage and more time, but a better product. LB is relatively new and is ineffective in resolving fragments larger than 5 kbp. However, with its low conductivity, a much higher voltage could be used, which means a shorter analysis time for routine electrophoresis. As low as 1 base pair size difference could be resolved in 3% agarose gel with an extremely low conductivity medium. Most SDS-page protein separations are performed using a discontinuous buffer system that significantly enhances the sharpness of the bands within the gel. During electrophoresis in a discontinuous gel system, an ion gradient is formed in the early stage of electrophoresis that causes all of the proteins to focus into a single sharp band in a process called isotachophoresis. Separation of the proteins by size is achieved in the lower, resolving region of the gel. The resolving gel typically has a much smaller pore size, which leads to a sieving effect that now determines the electrophoretic mobility of the proteins. Visualization. After the electrophoresis is complete, the molecules in the gel can be stained to make them visible. DNA may be visualized using iodine bromide which, when intercalated into DNA, fluoresce under ultraviolet light, while protein may be visualized using silver stain or Kumasi brilliant blue dye. Other methods may also be used to visualize the separation of the mixture's components on the gel. If the molecules to be separated contain radioactivity, for example in a DNA sequencing gel, an autoradiogram can be recorded of the gel. Photographs can be taken of gels, often using a gel dock system. Downstream processing. After separation, an additional separation method may then be used, such as isoelectric focusing or SDS-page. The gel will then be physically cut, and the protein complex is extracted from each portion separately. Each extract may then be analyzed, such as by peptide mass fingerprinting or de novo sequencing after in-gel digestion. This can provide a great deal of information about the identities of the proteins in a complex. Applications: estimation of the size of DNA molecules following restriction enzyme digestion, for example in restriction mapping of cloned DNA. Analysis of PCR products, for example in molecular genetic diagnosis or genetic fingerprinting, separation of restricted genomic DNA prior to southern transfer, or of RNA prior to northern transfer. Gel electrophoresis is used in forensics, molecular biology, genetics, microbiology and biochemistry. The results can be analyzed quantitatively by visualizing the gel with UV light and a gel imaging device. The image is recorded with a computer-operated camera, and the intensity of the band or spot of interest is measured and compared against standard or markers loaded on the same gel. The measurement and analysis are mostly done with specialized software. Depending on the type of analysis being performed, other techniques are often implemented in conjunction with the results of gel electrophoresis, providing a wide range of field-specific applications. Nucleic acids. In the case of nucleic acids, the direction of migration, from negative to positive electrodes, is due to the naturally occurring negative charge carried by their sugar phosphate backbone. Double-stranded DNA fragments naturally behave as long rods, so their migration through the gel is relative to their size or, for cyclic fragments, their radius of gyration. Circular DNA such as plasmids, however, may show multiple bands. The speed of migration may depend on whether it is relaxed or supercoiled. Single-stranded DNA or RNA tend to fold up into molecules with complex shapes and migrate through the gel in a complicated manner based on their tertiary structure. Therefore, agents that disrupt the hydrogen bonds, such as sodium hydroxide or formamide, are used to denature the nucleic acids and cause them to behave as long rods again. Gel electrophoresis of large DNA or RNA is usually done by Agarose gel electrophoresis. See the chain termination method page for an example of a polyacrylamide DNA sequencing gel. Characterization through ligand interaction of nucleic acids or fragments may be performed by mobility shift affinity electrophoresis. Electrophoresis of RNA samples can be used to check for genomic DNA contamination and also for RNA degradation. RNA from eukaryotic organisms shows distinct bands of 28s and 18s of RNA, the 28s band being approximately twice as intense as the 18s band. Degraded RNA has less sharply defined bands, has a smeared appearance, and intensity ratio is less than 2:1. Proteins. Proteins, unlike nucleic acids, can have varying charges and complex shapes, therefore they may not migrate into the polyacrylamide gel at similar rates, or at all, when placing a negative to positive EMF on the sample. Proteins therefore, are usually denatured in the presence of a detergent such as sodium dodecyl sulfate that coats the proteins with a negative charge. Generally, the amount of SDS bound is relative to the size of the protein, so that the resulting denatured proteins have an overall negative charge, and all the proteins have a similar charge-to-mass ratio. Since denatured proteins act like long rods instead of having a complex tertiary shape, the rate at which the resulting SDS-coated proteins migrate in the gel is relative only to its size and not its charge or shape. Proteins are usually analyzed by sodium dodecyl sulfate polyacrylamide gel electrophoresis, by native gel electrophoresis, by quantitative preparative native continuous polyacrylamide gel electrophoresis, or by 2D electrophoresis. Characterization through ligand interaction may be performed by electroblotting or by affinity electrophoresis in agarose or by capillary electrophoresis as for estimation of binding constants and determination of structural features like glycan content through lectin binding. History: 1930s first reports of the use of sucrose for gel electrophoresis, 1955 introduction of starch gels, mediocre separation, 1959 introduction of acrylamide gels. Disc electrophoresis. Accurate control of parameters such as pore size and stability. And, 1966 agar gels, 1969 introduction of denaturing agents especially SDS separation of protein subunit, 1970 Lamli separated 28 components of T4 phage using a stacking gel and SDS, 1972 Agarose gels with iodium bromide stain, 1975 two-dimensional gels. Isoelectric focusing then SDS gel electrophoresis, 1977 sequencing gels, 1983 pulsed field gel electrophoresis enables separation of large DNA molecules, 1983 introduction of capillary electrophoresis, 2001 standardized time of polymerization of phage gels enables clean and predictable separation of native proteins. A 1959 book on electrophoresis by Milan Beer cites references from the 1800s. However, Oliver Smithies made significant contributions. Beer states, the method of Smithies is finding wide application because of its unique separatory power. Taken in context, Beer clearly implies that Smithies' method is an improvement. See also: fast parallel proteolysis. References. External links: Biotechniques Laboratory Electrophoresis Demonstration, from the University of Utah's Genetic Science Learning Center, Discontinuous Native Protein Gel Electrophoresis, Drinking Straw Electrophoresis, How to Run a DNA or RNA Gel, Animation of Gel Analysis of DNA Restriction, Step-by-step photos of running a gel and extracting DNA, Atypical Method from Wike University, 2D Electrophoresis Principles and Methods Handbook.